Mitosis, the story of how one single cell becomes two identical cells. The cell cycle, also known as the cell division cycle. The process a cell goes through that takes place in a eukaryotic cell. It happens between formation and the second it replicates. There are two main parts, interphase and mitotic phase. Fertilized eggs become mature organisms during the cell cycle. It forms hair, skin, body cells, and some internal organs. Interphase. There are three main parts, G1 phase, S phase, and G2 phase. The cell is formed and functions normally. It's only defined by the absence of cell division. It obtains nutrients and duplicates its chromatids, which are genetic material. Chromatids are in the nucleus of the cell and made of molecule DNA. Parts of the chromatid are the centromere, short arm, and the long arm. Most time is spent in interphase by cells. About 90% of cells are in interphase right now. G0 phase, growth 0. Cells sometimes exit the cell cycle from G1 and enter G0. The cell is alive and metabolically active but does not divide. No DNA is copied and no preparation for cell division is done. If damaged, the cell cannot be replaced. It helps make the heart muscle, eyes, and brain cells. G1 phase, which is growth 1. The cell grows. New cells are identical daughter cells. This happens after cytokinesis. The cytoplasm divides during cytokinesis and new organisms are made. S phase, synthesis phase. It becomes to where each chromosome has two identical DNA double helix molecules. The chromatin holds duplicated chromosomes together and is copied during this phase. G2, growth 2. This is the final and shortest phase. A cell is in the G2 phase 4 to 5 hours. This is where rapid growth prepares the cell for mitosis. Karyokinesis, which is mitosis. After G2 phase, the cell is ready for division. The nucleus is what divides. The chromosomes are duplicated before mitosis, and cytokinesis follows mitosis. The cytoplasm is divided during cytokinesis, and identical daughter cells are made. A cell doesn't go through mitosis unless it has a nucleus, and bacteria uses binary fission to divide and reproduce. Body cells are called somatic cells, and reproductive cells are called germ cells. Mitosis, division of the nucleus. There are 4 phases: prophase, metaphase, anaphase, and telophase. In an animal cell, you divide cytoplasm by constricting the cell membrane in the middle to form a cleavage furrow to perform cytokinesis. In a plant cell, you form a cell plate in the center to divide cytoplasm to perform cytokinesis. Chromatin can be clearly visible or not clearly visible. DNA is shortened and condensed by coiling. DNA shortening and condensing by coiling makes the chromosomes become visible. The nuclear membrane is broken down in prophase. Enzymes help break down the nuclear membrane. The spindle fibers later attach and move chromosomes in prophase. The double chromosomes are held together by spindle fibers, and during metaphase, chromosomes on the equator line up on the equator or the middle of the cell. During anaphase, sister chromatids are separated and moved to opposite sides of the cell. And during telophase, chromosome pairs are pulled to opposite poles of the cell, spindle fibers disintegrate, and the nuclear envelope and nucleus reform before the chromatins uncoil. This is the story